The human eye is quite simply a masterpiece of biological engineering. It is our primary window to the world, a delicate, intricate organ that lets us perceive the beauty and complexity around us. To appreciate its capabilities, we must understand its fundamental components. Iris, the colorful ring that controls the pupil. Pupil, the small opening that widens or narrows to regulate incoming light. Lens, deeper inside, not rigid but flexible and crystalline. Accommodation, the lens changes shape to shift focus. Vitreous humor, the clear gel-like substance filling the eyeball. Every component works in perfect harmony, an elegant natural solution. The retina is the biological equivalent of a camera sensor but far more sophisticated. Here the magic begins. Photons are converted into the nervous system's electrical language. Optic nerve. This stream of signals is gathered and transmitted out of the eye. A one-way highway carrying constant visual information to the visual cortex. In the brain, these signals become the rich, detailed 3D images we call reality. The journey from a single photon to conscious sight is a breathtaking feat made possible by the seamless collaboration of these key parts. The retina is where the physical world of light is translated into the biological world of perception. This delicate, paper-thin membrane is packed with an incredible density of photoreceptor cells, specialized neurons that respond to light. The first type, the rods, are extraordinarily sensitive to light levels. They are responsible for our vision in dim conditions. The second type of photoreceptor cells are the cones. Cones are responsible for our perception of color and our ability to see fine details. They require much brighter light to function. There are 3 types of cones, each sensitive to a different wavelength of light. Red. The retina is not simply a passive sheet of these cells, it is a complex, multi-layered neural circuit. Other cells within the retina, such as bipolar and ganglion cells, begin to process these initial signals. They gather information from multiple photoreceptors, enhancing edges, detecting motion, compressing the data before it is even sent to the brain. This processed information is then channeled to the ganglion cells, whose long axons bundle together to form the optic nerve. When we attempt to quantify the resolution of the human eye in terms familiar to us from the world of digital cameras, the numbers are simply staggering. Scientists and engineers have made calculations based on the density of photoreceptor cells in the retina and the eye's ability to move and scan a scene. The most cited estimate suggests it would need to be around 576 megapixels. This number is almost incomprehensible when compared to the cameras we use every day. Even the most advanced professional digital cameras today typically offer resolutions between 50 and 100 megapixels. However, this comparison is not entirely straightforward. A digital camera captures an entire scene in a single static frame with uniform resolution across the entire image. Instead, it is a continuous stream of information with an incredibly high-resolution area at the center. Our brain then stitches these high-detail snapshots, taken as our eyes dart around, into a seamless and seemingly complete picture of our surroundings. This immense resolution allows us to perceive the world with breathtaking clarity. While the entire retina is a marvel, one small region stands out as the absolute pinnacle of visual acuity, the fovea. When you read a book, look someone in the eye, or focus on a tiny insect, you are using your fovea. Although it covers less than 1% of the retinal area, it is so crucial that nearly half of the brain's visual cortex is dedicated to processing information that comes from it. In this region, the retina is thinner because the overlying layers of cells and blood vessels are pushed aside, allowing light to fall directly onto the photoreceptors with minimal obstruction. Furthermore, the fovea is almost exclusively packed with cone cells, the photoreceptors responsible for high-resolution color vision. The density of cones here is astonishing, reaching up to 200,000 cells per square millimeter. This incredible density is what provides the foundation for our high-definition central view. This concentration of cones in the fovea is the reason why our vision is not uniformly sharp. You must move your eyes to bring new words into the foveal region to read them clearly. This is a fundamental aspect of how we see, constantly directing our foveal spotlight to the points of interest in our environment. This design, with a high acuity center and lower acuity periphery, is a brilliant compromise between detail and efficiency. Beyond its incredible resolution, the human visual system offers a remarkably wide field of view. With both eyes open and looking straight ahead, a healthy human can perceive a horizontal arc of nearly 180 degrees. This panoramic vista is crucial for our spatial awareness and our ability to navigate complex environments safely. This wide field of view is actually a composite image, a phenomenon that provides us with another extraordinary ability: stereoscopic vision. Each eye on its own has a field of view of about 120 to 130 degrees vertically and around 160 degrees horizontally. The field is limited on the inside by our nose. When the views from both eyes are combined, the central portion, approximately 120 degrees wide, is seen by both eyes simultaneously. This overlapping area is the basis for our depth perception. Because our eyes are set a few inches apart, each one captures a slightly different perspective of the world. This stereoscopic vision is what allows us to judge how far away an object is. It gives our world a sense of volume and space that a single eye or a single-lensed camera just cannot replicate. The human eye possesses an astonishing ability to adapt to a vast range of lighting conditions, a dynamic range that far surpasses any single digital camera. We can see, albeit with different levels of detail, the first line of defense is the pupil. In bright light, the iris constricts, shrinking the pupil to a diameter as small as 1 millimeter. This increase in aperture allows the eye to gather as much available light as possible, increasing its light-gathering capacity by a factor of more than 30. This mechanical adjustment is relatively quick, but it is only the first part of our adaptation to darkness. The more profound and time-consuming part involves a switch in the retina's primary workforce, from the cones to the rods. There are about 120 million rods in each retina, compared to only about 6 million cones. Once our eyes are dark adapted, a process that can take 20 to 30 minutes, they are so sensitive that under ideal conditions, a single rod cell can detect a single photon of light. The cones give us the vibrant, detailed world of daylight, while the rods provide a sensitive but colorless view of the nocturnal world. It is a fluid and elegant solution, allowing us to remain effective visual beings from dawn through to the deepest night. Our perception of color is one of the most wondrous aspects of vision, transforming a monochrome world of shapes and shadows. This ability originates with the 3 types of cone cells in our retina. Each type contains a different photopigment called photopsin, which is tuned to be most sensitive to a specific range of light wavelengths. Chart: absorption spectra of the 3 cone types, SML, against the visible spectrum. Short wavelength cones, most sensitive to blue. Medium wavelength cones, most sensitive to green. Long wavelength cones, most sensitive to red. Every color we see is a result of the brain interpreting the relative strengths of the signals coming from these 3 types of cones. Similarly, a purple flower stimulates the red and blue cones, while a turquoise object stimulates the green and blue cones. It is a system of combinatorial coding, where an almost infinite variety of colors can be perceived from the graded responses of just 3 inputs. It is estimated that the average human can distinguish between 1 and 10 million distinct shades of color. This sensitivity lets us discern the subtle difference between a ripe and unripe fruit, perfect for a primate species that evolved to find colorful fruits and leaves in the dappled light of a forest canopy. When we place the human eye alongside a modern digital camera, the comparison reveals fascinating differences in design philosophy. A digital camera is a marvel of precision engineering, built for a singular purpose: to capture a static, high-resolution image with as much fidelity as possible. It features a rigid, perfectly ground glass lens, a flat, uniform sensor, and a mechanical shutter. The human eye, by contrast, is a product of biological evolution, and its design is one of brilliant compromise and dynamic adaptation. Its lens is soft and flexible, not perfectly shaped. The retina performs significant processing right on the retina itself, enhancing edges and detecting motion before the signal even leaves the eye. The brain then completes the job, correcting for the lens's imperfections and filling in the blind spot where the optic nerve is. Consider the task of focusing. A high-end camera uses a complex autofocus system with motors and sensors. The human eye focuses by subtly changing the shape of its own lens using the ciliary muscle. This process is incredibly fast, silent, and efficient, allowing us to shift our gaze from near to far in a fraction of a second without conscious thought. Furthermore, a camera captures an image and then a separate computer processes it. The eye performs significant processing right on the retina itself, enhancing edges, detecting motion, and constructing a stable 3-dimensional world from 2 constantly moving imperfect retinal images. A camera records a scene, our visual system creates an experience. It is the difference between a tool and a living, integrated part of our own consciousness. Beyond the headline features of resolution, color, the human eye possesses a suite of extraordinary abilities that often go unnoticed. One of the most remarkable is its speed of focus. We can change our focal point in as little as 200 milliseconds. Another astonishing feat is the eye's ultimate sensitivity. In complete darkness, the human eye can detect a flash containing as few as 5 to 10 photons. Our visual system thus operates at the quantum level, registering single packets of light. This sensitivity is a relic of our evolutionary past, a vital advantage for ancestors without artificial light. Perhaps most fascinating are the movements our eyes make without our awareness. They constantly perform tiny, involuntary microsaccades. They shift the image across photoreceptors, preventing receptor adaptation and shutdown. Without them, a static scene would fade within seconds. Instantaneous focus, quantum-level sensitivity, the ceaseless jitter of microsaccades all combine to make our sight robust and effortless.